Hello there, my name is Caroline Edo, and I will be presenting the paper on Analgesia mediated by the TRPM8 called receptor in chronic neuropathic pain. Chronic pain and analgesia. Chronic pain can be hyperalgesia, which is abnormal responses to pain, or allodynia responses to normally harmless pain, or even spontaneous pain. Sensitization as a result of changes to damaged or undamaged primary afferent neurons show a serious clinical problem with few therapeutic options. Now, a mechanism of analgesia acting as cutaneous cooling may prevent this pain stimulation. A quick research background. Sensations such as pain, including chronic pain and touch, are moderated by peripheral sensory neurons. Temperature does interact with pain. Ion channels of the transient potential family function as thermal investigators by supplying the receptor potentials that initiate signaling to the central nervous system. Examples are TRPM8, TRPV, and TRPA. TRPM8 expresses menthol in cold-evoked responses in sensory neurons, while TRPV receptors will open in response to noxious heat, thus allowing an influx of calcium ions in the nervous system. Additionally, TRPA1, a cold receptor, seems to elicit reflex pain behaviors and increases thermal and mechanical responsiveness in the neuropathic state. Researchers in this experiment seek to know if TRPM8 in a subpopulation of sensory afferent specific pharmacological cooling agents can elicit analgesia in neuropathic and other chronic pain. They actually use TRPM8 activators such as isolene or menthol. Their target to inhibit the characteristic sensitization of dorsal horn neurons and behavioral reflex facilitation in rat models with neuropathic or chronic pain. Researchers in this experiment topically applied isolene, a TRPM8 activator, in the pores of a rat with sciatic nerve injury in a 1 centimeter deep bedrock solution kept at 30 degrees Celsius to avoid any effect on the local skin for 5 minutes. Secondly, the presence of TRPM8 was investigated in the dorsal root ganglion and spinal cord using immunoblotting and immunochemistry. The result of this experiment shows a reversal of nerve injury-induced reflex sensitization by peripheral activation of TRPM8 channel receptors to reduce neuropathic pain. This is consistent with a TRPM8 mediated mechanism. Poor emission at 60 degrees Celsius to 20 degrees Celsius for 5 minutes also produce statistically significant mechanical analgesia. We also see that activating TRPM8 in a cell population of afferent by isolene, menthol, or moderate cooling shows release of glutamate through inhibitory group 2 and 3 M-GLU receptors to reduce neuropathic pain. Now, the precise identity of the TRPM8 containing co-responsive afferent is still not clear. The analgesic effect of isolene were only seen in the sensitized pain state, but were not restricted to nerve injury because sensitization due to peripheral inflammation, afferent demyelination, and TRPA1 activation was also reduced. Thus, further research is still needed. However, TRPM8 can also be reversed by administering group 1 and 2 M-GLU antagonists, therefore reversing the inhibitory effect of isolene on sensitized neurons. In conclusion, we see the topical application of TRPM8 activators reduce pain by producing analgesic effects. It is shown through the reversal of sensitization in behavioral reflexes. For further experiments, Perhaps could TRPM8 be a possible solution for psychogenic pain? We never know. Thank you for listening.